Animal experimentation, also known as vivisection, has been the subject of intense debate for hundreds of years. Each year, 100 to 200 million non-consenting animals are used and killed in the name of science. There are countless examples of gut-wrenching cruelty resulting from the inevitable consequence of using these creatures as objects, as means to an end, with no more consideration given to their interests as one would give to a centrifuge or a pipette. Kittens' eyes have been sewn shut and dogs electrocuted into states of despair, euphemistically called learned helplessness. PTSD has been induced in monkeys by subjecting them to months of food deprivation, endless noise, and extreme physical confinement. Guinea pigs have been injected with toxic nerve agents, sheep with formaldehyde solutions, and mice with a battery of toxic chemicals. If you find these examples abhorrent, you are not alone, as half of Americans oppose the practice. But what does the average person really know? Surely scientists, being among the most intelligent and well-educated ranks of society, must have compelling evidence and strong ethical justifications to uphold this tradition. Like all traditions, though, vivisection must be scrutinized to determine whether it is fruitful to continue the practice. It is important to note that scientists are not ethicists, and given the fact that animal experimentation is institutionalized, its efficacy is seldom questioned. As important as they are, ethical arguments will be set aside, and this video will focus on the empirical evidence. Does animal experimentation work? Is it scientifically justified? Let the evidence speak for itself. If animal models work, they must have a dependable predictive power. However, the chances of a new drug passing through all clinical trials to market is very low. 96% of drugs shown to be safe and effective in animal models never make it to market. When withdrawals, duplicates, and low patient response rates are considered, 99% of all novel drugs fail. 3 examples will illustrate how poorly success in animal models translates to success in humans. Lou Gehrig's disease, or ALS, is a neurodegenerative disease that causes muscle stiffness, weakness, and sometimes dementia. Out of 100 potential drugs that worked in mice, only 1 was found beneficial in humans. In another case, a grand total of 2 out of 500 neuroprotective interventions working against stroke in mice were successful in humans. Since mice share about 85% of our DNA with us, non-human primates who can share up to 98% are thought to be better models. Unfortunately, while better approximations, they don't prove accurate enough. For example, 90 HIV vaccines that worked in non-human primates all failed in preventing HIV in human trials, and some are even thought to have increased the chances of contracting the disease. Advocates of animal experimentation rationalize these numbers by claiming that drug development is trial and error, and that the majority of compounds tested in humans also fail. While this gives some context, it does not justify using a potentially flawed methodology. Therefore, we must examine if animal models are in concordance with human outcomes. The preponderance of evidence shows that animal models predict human outcomes accurately in only a minority of cases. An umbrella study of 20 systematic reviews covering a wide range of research fields found that only 2 reviews demonstrated that animal models had a significant potential to contribute toward clinical interventions in humans. However, one of these reviews was contentious and is under debate. It is important to note that these papers were published in top journals, were heavily cited, and included studies involving chimpanzees. This demonstrates once again that even our closest cousins are not similar enough to us to be reliable models. A study analyzing 100 abstracts found that none stated unequivocal and conclusive concordance between animals and humans. Only 20% described a limited concordance, while the vast majority, 75%, found that human responses from animal data was significantly limited. 5% even flat out admitted that animal experiments could have been entirely omitted and carried out more effectively in clinical or observational studies. Animal models have performed no better in other studies. A systematic review of interventions to treat head injury, respiratory distress syndrome, osteoporosis, stroke, and hemorrhage found animal and human results were in concordance only 50% of the time. Why use animals when we'd have the same predictive power by flipping a coin? Even chimpanzee experiments, which are purported to be the gold standard of animal models, found that out of 76 experiments, only 28 cases, or 37%, were replicated in humans. These were major papers published in top journals, with each being cited over 500 times. Concerning toxicology, animal models have fared no better. 7 systematic reviews examining the predictability of toxicological outcomes failed to demonstrate any utility by utilizing animal models. Using animal models to predict how humans will react to chemicals that may cause birth defects has also proven to be futile. Of 3,100 chemicals known to cause birth defects, 1,500 produce birth defects in animals but not in humans. The utility of animal models predicting birth defects in humans was staggeringly low, with a mere 40 chemicals, or 3%, causing birth defects in both animals and humans. This analysis shows that animal models fail in 2 respects. They result in many false positives, which are in this case drugs that are toxic in animals but safe in humans. They also fail in predicting true positives, that is, drugs that are toxic in both animals and humans. When examining drug side effects, animal models also prove to be poor in predicting human reactions to drugs. A study of 6 drugs, of which animals and humans share 22 side effects, showed that the side effects present in animals were present in humans only 31% of the time. This again demonstrates that animal tests can mislead researchers by flagging drugs as unsafe when they're not, or missing real risks to humans entirely. Taken together, these findings raise extraordinary doubts about whether animal experimentation is a reliable tool for medical progress. As shown, animal results are inadequate and often misleading when applied to humans. It is worth giving a brief explanation as to why animals are such poor models. Animals and humans are similar in many respects. We both share traits that are essential to our survival. For instance, glycolysis in mice and humans will be identical because a mutation in any genes involved in the breakdown of sugar will be lethal. The same is true for certain aspects of behavior, such as aversion to harmful and threatening stimuli. However, there are key differences that pose insurmountable obstacles to accurately applying results from animals to humans. Scientists who advocate for animal experimentation act as if mice are miniature humans. They conveniently forget we differ from animals in immutable aspects, such as our metabolism, physiology, gene expression, and pharmacokinetics. In terms of gene expression, this should be almost self-evident. By just looking at a mouse and a human, one can see the result of 75 million years of genetic separation. Genetics and epigenetics are the key players in explaining why mice are mice and humans are humans. Even though mice share 85% of our DNA with us, gene expression makes all the difference in determining how an organism will develop. To illustrate this, we can use the analogy of 2 different pianos. We can be generous to proponents of vivisection and for the sake of argument, assume we have the same genes as mice do, just like 2 pianos have identical sets of keys. What matters is how these genes are expressed, or in the analogy, in what order the piano keys are played. Holding down different keys for varying amounts of time and in different orders produce different melodies. One sequence could result in a piece by Shostakovich, while another could play something by the Sex Pistols. This is analogous to how genes can be turned on or off and expressed for differing amounts of time, resulting in 2 distinct organisms. Examples of how genetics complicate things can be easily demonstrated. Even in humans, a difference in a single nucleotide can significantly impact an organism's phenotype. Take sickle cell anemia, which is the result of a single point mutation. One allele gives resistance to malaria at the expense of a shorter lifespan, showing that even small changes in genetics can cause important phenotypical differences. Dissimilarities in genetics can produce radically different outcomes in different strains of the same species. For instance, the same genetic mutation that causes Fragile X syndrome in different strains of mice produces different behaviors. Another example is that mice of different strains recover differently from spinal cord injuries when given the same drugs. Additionally, one study could not predict how mice of 11 different strains would respond to pain by measuring 12 different behavioral measures. Amazingly, even mice of the same strain bought from different suppliers have produced varying experimental outcomes. Uncontrollable differences in laboratory environments can influence experimental outcomes in unforeseeable ways. Handling, restraint, stressful administration of compounds, and environmental stressors can all impact an experiment's results. These factors can alter physiology, hormones, and immune and behavioral responses. Just being confined to a cage has been shown to produce abnormal behavior in the lab animals. 50% of mice and 90% of monkeys display abnormal behavior in captivity, casting doubt on how useful the resulting behavioral data will be. The environment also plays a significant role. Genetically modified mice bred to develop heart defects had those defects disappear when given a bigger cage. Even the type of flooring in animal housing affects whether a drug will be effective. Noise level in the labs have also been shown to damage blood vessels in animals. Controlling for these factors poses another significant hurdle vivisectors must take into account. Animal studies have been shown to have prevailing technical problems that are not taken into consideration by most scientists. One major form is selection bias. Researchers often select mice they perceive to be more robust for the experimental group instead of the control. Unintended influences such as these can result in differing outcomes. There are various methods to control for selection bias, such as the use of random number generators. However, scientists rarely put these controls into use, as demonstrated by a study that showed that only 12% of papers use randomized allocation. Observational bias is also endemic to animal experiments. Observation bias is caused by researchers unconsciously evaluating experimental groups in ways that might suit their interests. Any scientist can attest that one of the hardest parts of research is spending months on a project resulting in no publishable data. To avoid these altered evaluations, researchers can examine their results without knowing which animals are in the control or experimental group. Just like selection bias, observational bias controls are used in a minority of cases, as evidenced by a study showing that they are employed in only 14% of papers. When I first began studying science, I had a rosy, naive view of what it would be like to be around those in my chosen profession. Scientists, I thought, must be more rational, kinder at heart, and more open to new ideas than the average person. However, I soon learned that scientists have the same character faults that all humans have. I've seen peers studying to become medical doctors openly mock others with mental illnesses. I've seen people with doctoral degrees behave like children, making bigoted remarks and bullying their co-workers. Vivisectors have based their whole careers on experimenting on animals. Imagine the discomfort they must feel when presented with data questioning the utility of their life's work. What would it mean if all their years of study, long hours in the lab, and personal sacrifice were for naught? The truth is that being intelligent or well-educated does not immunize anyone against self-interest or denial. In science, as in all human endeavors, it takes extraordinary courage to question the foundations of one's own beliefs, to risk admitting that the path well-traveled may be a dead end. Progress demands not blind loyalty to tradition, but an unwavering commitment to reality, no matter how uncomfortable it may be. It is not enough to proclaim science as self-correcting. We must be willing to correct it. And when tradition stands in the way of compassion, of truth, and of better ways forward, we must have the strength to inter it in the ground where it belongs.